Hey everybody, so we're going to look at operons, just a real quick look at a couple of diagrams. Here we see the lac operon, which is a mechanism for controlling gene expression found in prokaryotic organisms. So in this instance, we are looking at a strand of DNA across the middle here, and then different regions of that DNA that are known as the cap site, the promoter, the operator, the lac Z, lac Y, and lac A. Now the lac Z, Y, and A are actually the three genes that are responsible for the production of lactase and the associated features of maintenance of lactose inside of this prokaryotic organism. So these are like the three functional genes that are going to handle the presence of lactose. The operator is a region of DNA with a particular code that therefore has a particular shape that allows it to associate with a separate protein known as a repressor. Now, a repressor, like the name implies, represses the expression of the gene, and it does that by just physically getting in the way of the RNA polymerase. So the RNA polymerase is able to attach at the promoter, which is then going to begin the process of transcription, where we're going to have that unwinding with the help of some helicases and the transcription of the genes. However, the repressor, if it's physically blocking the RNA polymerase, prevents that from happening. So in the case of lactose in a lac operon system, the lack of lactose causes the repressor to have a shape that enables it to be active, which is to say the repressor naturally, in the absence of lactose, has a particular shape that allows it to functionally block the transcription of the lactase genes. Therefore, when we don't have lactose, the repressor is active and therefore blocking the production of lactase, which makes sense because we don't have any lactose. When there is lactose, the lactose, or a product of the excess lactose that's in the cell, there's going to be some sort of chemical equilibrium here, and so we have allolactose, which is a molecule that's going to be in higher abundance in the presence of lactose, is going to cause a change in shape of the repressor and pull it away from that operator. So just like all the chemical interactions we've seen, hydrogen bonds pulling on things, shape changes, it no longer binds to the operator, and therefore RNA polymerase is able to continue on transcribing these genes and leading to the production of lactase, and again associated other factors that are going to then start breaking down that lactose until the lactose concentration is low enough that the repressors are largely active again, and then it hits the brakes on that system. So bringing in lactose leads to the production of lactase. No more lactose shuts down the production of lactase, increasing efficiency, so we're not just mass producing lactase when we don't need it, we being this prokaryotic organism. Now the cap site's an interesting thing, sort of a separate layer on, or an extra layer on top of all this, where the cap site is a additional promoting factor that is active when there is a low concentration of glucose. And the reason why that's significant is because what the lactose is getting broken down into is galactose and glucose. So the lack of glucose would indicate that there's an insufficient breaking down of lactose, and therefore would promote additional lactase production. So it's sort of like an extra booster step. So that way, if we have a presence of lactose and we have low glucose, then we're going to have sort of twice the pressure trying to drive this forward to produce the lactase. Meanwhile, the trip operon is sort of the same thing, just a little bit in reverse. It's the same general thing. We have a promoter region, we have the operator region, a repressor which can block that operator, and then we have, in this case, 5 different TRP-related genes. Interestingly, tryptophan is an amino acid. And so when we talk about the production of tryptophan from this, you have to remember it's an amino acid. It's not a protein. It's literally one of the subunits of protein. So the reason why there's so many genes here is that there's a whole host of proteins that are needed to basically step-by-step create tryptophan, the amino acid. So anyway, the process here is that the repressor is by default inactive, which means that we're just running and we're making tryptophan, But when we have an abundance of tryptophan, it then binds to the repressor, changing its shape and activating the repressor, therefore hitting the brakes on the production of tryptophan. So exactly the same overall mechanism as what we saw with the lac operon, but the repressor is sort of working in reverse. The repressor is inactive by default for tryptophan, but the repressor is active by default with regards to lactose. So there's a little bit about operons. Hope that's helpful.